Well, thank you all for coming to this. Let's see. Alex Martin gave me instructions on what to say. The 15th reunion of CAOS, is it correct? 15th year. 15th year, there was some time off. Right, 15th edition. Now, from this exchange, you have an idea of how this committee functions. There's not one idea that is shared across the small group of people. And that makes for lots of fun meetings, because we schedule 10 minutes or something, and 2 hours later we're still saying, what are we supposed to be talking about? And things go. In any case, we don't want to have the same kind of uninformed discussions during our presentations. One of the beautiful features of CAOS, so those of you already have been here, I know, is that we have a fairly lengthy period for question asking, about 30 minutes. So the speakers will have 45 minutes, and whoever is chairing that session will, at 45 minutes exactly, unplug the mic so that you have to stop. And this makes sure that we have 30 minutes for discussion. So thank you all for coming. Let me welcome all of you on behalf of the organizing committee and our, especially the two people who did most of the work, Stefania Bracci and Moritz Wurm. I have a couple of announcements to make. The poster sessions will go on both days, and they are open the whole time, so please go to the poster sessions during the breaks. And we've also scheduled for two presentations, abstract presentation winners. One other announcement I have to make: those of you who use X or Twitter, the hashtag is CAOS, C-A-O-S 2024. So please, if you have photos or papers you like or whatever you want to communicate, use the hashtag CAOS 2024. A couple of other announcements. The dinner, social dinner, is tomorrow evening at 8 o'clock in Hotel Rovereto. If everyone knows where Hotel Rovereto is, it's in the main road towards the station, near the end of that road. Let's see, poster sessions, hashtag Twitter 50. Okay, I've read all the instructions that I was given, so I'm going to start this morning's session. The first presentation is by Luca Bonini from the University of Parma. And the title of his presentation is Objects, Actions, and Beyond, Neural Basis of Pragmatic Remapping. I'm really curious about the meaning of beyond. So thank you all for coming. Luca.
Can you hear me? Yes. Okay. Okay, so thank you very much for the invitation. It has been quite a trouble for me to be here today, but I really wanted to be here. And it's a pleasure because this workshop deals with a topic that I've been working on since my PhD, and especially from a neurophysiological perspective. I dealt with object and actions. Yes, probably. Okay. I dealt with the topic of object and actions from a neurophysiological perspective, starting from some of the original background provided by research started in the 90s, so in the last year of the previous century, where neurophysiology in non-human primates demonstrated the existence of a variety of neuronal categories. Categories are reassuring because being able to subdivide the complex set of things into categories gives the feeling to be able to make order, to organize, ultimately to understand the complex system as the brain certainly is. But there was somehow from the tradition of ecological psychology the idea that categories could be used or should mean something specific, and in the motor system there were evidence of the presence of neurons encoding visual and sensory information. According to Gibson, the visual world has the appearance of inviting many kinds of behavior, and the neurophysiological properties that emerge from the recordings in the premotor cortex of non-human primates support this view by showing that, for example, there was a class of cells responding to the visual presentation of object and sustaining this activity until the monkey grasped actively the object, with the selectivity that remained from the visual presentation to motor execution. This was thought to be what you would expect from a neuron involved in the sensory motor transformation, so they call this neuron canonical because of this reason. Actually, subsequent evidence that I will provide suggests that this was not so canonical, actually. Another set of cells was the peripersonal neurons that are really fascinating. I will not have the time to include a discussion in my talk of these cells today, but they encode in a bimodal fashion a certain body part in somatosensory terms, and the somatosensory field expands in the visual space when object approach or detach from the body place field. So basically they provide a sort of representation, three-dimensional representation of the space centered on the body. Even in this case, we are in a premotor region here in blue, area 4, that actually subserve the control of defensive movement and axial body movement that enable the body to escape from stimuli approaching to it. So another example of the intrusion of sensory information into the motor system. The final class of cells is the famous class of mirror neurons that have been called in this way because the researcher found cells discharging during both the execution of an action and also when the monkey observed another agent perform a similar action, as if the observed actions mirror reflected onto the motor repertoire of the observer. So the research in the subsequent years showed that things are a bit more complex than that. In recent years, from the Hans Scherberger group in the DPZ in Göttingen, it was shown that by presenting monkeys with a large variety of objects of different size and shape, it was possible to identify in the anterior intraparietal area of the inferior parietal lobule a clusterization of objects based on the neuronal activity that segregate different object population that you can see here in different color based on their shape and three-dimensional properties. Which means that the visual processing of objects in AIP takes place essentially for extracting information that have to deal with the intrinsic properties of the object. While the technology that this researcher could apply enabled to simultaneously investigate with the floating microreactor arrays the neuronal activity both in the anterior parietal area, in the premotor cortex, and in the primary motor cortex, making a sort of dynamic picture of the sensory motor transformation while it occurred. And in the premotor cortex, as you will see here, this is an array with the firing neuron. This is the reconstruction of the monkey's kinematic during grasping. And as compared to AIP, they found that the neuronal activity in ventral premotor area 5 clusterized objects not based on their shape, but primarily based on the way in which the hand was shaped according to the object. And finally, in the primary motor cortex, the same cells recorded along the task unfolding did not discharge during object presentation of movement preparation, but during the actual execution of the end. So they basically provide an elegant picture of the sensory motor transformation in which AIP encodes the visual shape and object visual features relevant for grasping, Area 5 encodes the end shape during planning and execution of grasping acts, and finally Area 1 encodes the movement of the end for actively grasp the object. And these data fit well with the previous evidence that inactivation of area IP and the 5 impairs the visual motor transformation, affecting the end shaping before and during the approach of the end to the object. So the new technology actually enabled a very refined description and characterization of a process that take place not just because of a single neuron operating the visual motor transformation from object presentation to grasping, but because this kind of constancy in the representation of the ongoing motor plan from the visual description of the object to its acquisition with the end take place along a circuit that involves several distinct areas. Indeed, this is an aspect that in this paper has not been emphasized enough, but from also personal communication, I know that things are like this. If you check the single neuron in each of these areas, it is not so canonical because it suddenly changed its selectivity from object presentation to grasping. The stability is maintained within the circuit. So you cannot look at the single neuron and categorizing the neuron pretending to understand what the whole circuit is doing, because much of the information is emerging through the transfer from one area, from one step to the other. Beside this refined and more precise description, this is an elegant evidence of an idea that nevertheless was at least 20 years old, because this is a seminal paper by a group of authors, 𝗟𝘂𝗰 𝗕ន់, and Sakata, where they talked about cortical mechanism of visual motor transformation. And as you can see from this picture taken from this review, they pointed the spotlight exactly in this same circuit of areas that Scherberger has most recently investigated. So it was already, this is just to say that at the end of the 90s was already clear that there was a circuit in which visual information from the parietal cortex was conveyed to the ventral premotor cortex, and this was the basis for a visual motor transformation that is then demonstrated in many other studies in humans through causal inactivation, intracortical microstimulation, and so on and so forth. What is puzzling is that when moving from object to action, there has been somehow a sort of shift in the approach to the interpretation of the findings. Because when we deal with mirror neurons, we have cells that fire during action execution, and that also respond during the observation of an action performed by another individual. But in the interpretation of these cells, from affordance, that was a very sound and reasonable interpretation of the evidence concerning sensory motor processing of object, they switched to a completely different hypothesized function, which is action understanding. I'm quite skeptical and critical on the term action understanding in general, because I really don't know what understanding should mean. And whatever it means, if you try to investigate action understanding in operational terms, trying to invent a test as someone has done actually, you will always end up with transforming understanding into classification, recognition, match to sample, discrimination, something more simple that is not really understanding. Understanding is a concept that has been used in the language domain, that was suggested by the need to somehow interpret these findings in the light of the Lieberman theory of modern theory of speech perception, but it is difficult to fit with the evidence of such a mechanism in the monkey that does not imitate And according to the original interpretation of 𝗟𝘂, this kind of mechanism could be ideal for imitation, as it is then demonstrated actually in human. But if you look at the outcome of this original discovery by just taking the 5 seminal paper on monkey mirror neurons and looking at the cited by of this paper, this is the picture that comes out. So most of the study deal with the field of social cognition that has been certainly shaped considerably by this discovery, and most have to do with this concept of action understanding as well as imitation, that in human is a function reasonably attributed to this system, ranging from neuroimaging to TMS, EEG, and so on in terms of the paradigm and approach used to investigate this topic. So if we look at the original paper that described this cell in the premotor cortex, we see that the definition of mirror neurons claimed that mirror neurons require an interaction between the agent of the action and the object of it. The agent alone or the object alone were not effective. And in most mirror neurons, there was a clear relationship between the visual action they responded to and the model response they encoded. So basically 3 main features. The end alone is not effective, the object alone is not effective, the action is required. And there is also the need of a clear relationship between the visual and the model response, so in terms of visual motor congruence. What did we acquire in terms of knowledge from the subsequent studies? Well, this is a paper by Papadourakis and Rouse published in 2017, where it is clear that these neurons respond to the observation of action performed by the experimenter on 6 different objects. This is the preferred response, the one that is associated to the object evoking the highest discharge. This one is the response of action observation on the object that evoked the less preferred discharge, and this is the response to pantomimed action, so with no object. It is clear that the response is slightly lower, sometimes even not lower than the non-preferred transitive one, but basically all neurons with these properties respond also to intransitive actions. So the end alone is actually effective. Concerning the object alone, well, we devised a paradigm that I will show you better later, in which we could test action observation and action execution responses in different space, and the task enabled us to separate object presentation from action execution, and as in the classical canonical neuron task. But we could test these cells not only during the execution task done by the monkey. As you can see here, all these neurons have a response during the object presentation and then during the action execution, as you would expect from a canonical neuron. But many of these cells, more than 1/3 of the cells in the premotor cortex, that exhibited responses to action observation, either in the peripersonal or extrapersonal space, did also respond to the object alone. So the object alone very often is indeed effective. The last point was about the congruency. So the selectivity of the neuron for a given grip type performed by the monkey on an object should match the selectivity during action observation. Here you can see a recent paper published in 2019, again by the group of Vasily Strauss, where they tested 6 different objects during action execution by the monkey, and there was a nice selectivity. But if you try to rank the action observation response performed by the experimenter on the same object, you will see that there is no selectivity, which means that what these neurons are doing in the motor domain is different in terms of selectivity with respect to what they are doing in the visual domain. And nothing changes if you try to sort the neuron the opposite way. So you rank the neuron based on observation, and you try to see what happens if you keep the same ranking during execution. No selectivity, no match. The match is actually at chance level, because if you take 6 objects and neurons can be selective both during the execution and observation for one of these 6 objects, there is a specific probability that you can easily calculate, and this is exactly the number, the fraction of neuron that seems to match the visual and the model description. So it's a chance level, basically. Someone in the Peter Thier group recently wondered whether this could be because actually the match and the congruence is not at the level of the grip type, but is more generally at the level of the action goal. So they devised a test in which the monkey had to perform 3 different actions on the same object and then tested the possible visual motor congruence in this way. The result did not change. Most of the neurons, even analyzing this effect along the task unfolding period, are incongruent and shift their tuning from the motor modality to the visual modality during the task. So there is basically not a clear relation between the visual and the motor response, neither in terms of grip type nor in terms of action goal. So if you want to take together these criteria as originally proposed in the first seminal mirror neuron paper, according to the original definition, you would end up with an empty set of cells. So mirror neuron would not exist. Whereas they certainly exist. They have been described in many laboratories all over the world. Several people have seen these cells, and they could describe how they fire, but emphasizing different properties and a more complex relationship of these cells and of the motor system as a whole with the sensory information coming from the outside world. And this is because brain tends to mix. We have seen this in many domains, from many studies, from many laboratories, and there should be a reason why they do that. So I had this impression since the end of my PhD that things could be so complex. So we tried to exploit some new technological refinement that we could set up in our and other laboratories, so to record neuronal activity much longer than it was originally possible when these studies, those of canonical neuron, mirror neuron, were done. So using chronic electrodes or laminar probes that could enable us to sample in a completely unbiased way several cells simultaneously for a longer time to be able to test motor responses and object presentation responses, and in the same cells, randomly interleaved in the same session, also action observation responses done from a subjective viewpoint, from a third-person perspective, within the peripersonal space, in the extrapersonal space, so to be able to investigate the larger variety of variables at the single neuron level. And we did this in different areas of the so-called lateral grasping network. In this first study that I've already shown before, we could provide evidence of mixed selectivity in area 5. So most neurons in area 5 did actually encode both visually presented object, observed action, with different selectivity for the space sector in which the action of the experimenter should be presented to evoke the best response of individual neurons. We then moved to area F6 because from neuroanatomical studies we know that area F6, the pre-supplementary motor cortex, is an important source of projection and exchanges of information with the ventral premotor area F5. We used the same type of task and we could find evidence of action-related neuron and object-related neurons as previously described in the ventral premotor cortex. And the interesting thing is that when looking at the agent selectivity, so who was the agent of the action encoded by the neuron, we found some neurons that could be classified as self-type, so let's say motor, because they responded just when the monkey in blue was performing the action and not during the observation of the action performed by the experimenter. Another set of neurons that we could classify as other type because they responded just during the observation of the action performed by another, not when the monkey did the action itself. And another set of neurons again with both types of response. You could see here that this neuron did not respond only to the action execution but also to the presentation of the target. Indeed, by looking at this specific epoch, the object presentation epoch, we could find evidence of agent selectivity also during object presentation. So some neurons encoded the object as a target for the monkey alone, other just when the object was a target for the experimenter, and another set when the object was a target for both the experimenter and the monkey, as you would expect from a neuron that simply encodes the object presented at a certain distance from the animal. By focusing on this specific class of neurons, we found that actually it was more supported by the evidence to talk about mirroring mechanisms for the object rather than for the action, because this object basically afforded a potential motor action. But in their information, both with the coding analysis and more specific comparison of the firing features and firing properties of the cells, we could identify that there was a high similarity in their response when the object was presented as a target for the monkey or for the experimenter, but nevertheless it was possible to discriminate and decode who would have been the agent supposed to act on that object since the phase of the visual presentation. This is the reason why we propose this concept of object mirroring, because the affordance provided by the object could be modulated by the agent that was supposed to act on it. Moving to the supposed source of visual information to the wall circuit, area AIP, we recently published this paper in which we collected several individual neurons' activity simultaneously from a chronically implanted probe, and this enabled us to test a large variety of tasks, including all those that I've previously mentioned, and in addition also filmed action, static object, images of static hand, pantomiming or grasping object, and the static presentation of object or hands in the screen. What we found in this way is that there is a huge mixed selectivity of different types of information even in IAP. So we cannot find segregated neuronal category one dealing with a single aspect of the story, either object, action, or planning, grasping execution. But mixed selectivity was not random. So we could find from the baseline to action execution, and particularly during the object presentation phase, a reasonable grouping of the task condition solely based on the analysis of neuronal activity that treated together and emphasized the similarity between specific spaces in which this information were provided. So space seems to be a gating factor to emphasize and shape neuronal activity. When we looked more carefully at this aspect by basically focusing on all those neurons in area AIP that responded to the visual presentation of an object by this specific manipulation, we found a very interesting finding. So, interposing a transparent plastic barrier between the monkey and the object does not change absolutely anything in the visual features, metric features, distance of the object relative to the monkey. So, from a sensory point of view, the object is exactly the same, the condition is exactly the same, nothing changed. But in AIP in particular, more than half of the neurons encode a pragmatic representation of object, which means that if you present the object, as neuron 1 and neuron 2 elegantly show, during the visual motor task, the neuron responds, sometimes with a clear-cut selectivity for an object. It does so even when the monkey is instructed just to observe the object, not to grasp it during a no-go condition. But if you interpose the barrier, the monkey is still exactly as in this condition. The object is the same, nothing changed from the visual point of view, but the response is absent. So this set of neurons that are highlighted in light blue here in the picture seems to encode the object from a pragmatic point of view just when they can be the target of a subsequent action. If you look at this confusion matrix, the set of neurons that can be characterized as pragmatic, characterized by a pragmatic coding of object, they cluster the space, so the space of the visual motor task, and treat separately the condition in which the barrier was used, regardless of whether the target was a small or a big object, so regardless of the grip or perceptual features of the object. The other set of neurons, the 44% of the cells that we found in AIP, had instead a visual representation. And here, the clustering tend to separate primarily the object based on their shape, small on one side and big on the other side, and just after to segregate the different task conditions. So they don't have a pragmatic but a visual description of the object. We did this same task with the barrier also in all the other areas and found even more marked result. In F5, in general, most of the neurons tend to be reduced in their firing rate by the interposition of the barrier, and certainly all of them lost the selectivity for the object. Similar findings have been reported also in the pre-supplementary motor cortex. Here you have a nice response and selectivity for one of 3 different target objects. This is slightly reduced in F6 during the no-go condition because the monkey has not to act on this object, but the selectivity is completely abolished when the barrier is interposed. This same effect, not surprisingly, but actually this has been a sort of a paradigmatic change in the interpretation of action-related responses, was demonstrated in 2009 by a collaboration between the Parmas group and the group of Peter Thier when studying mirror neurons in the premotor cortex, so representation of action in this case. What happens if you test observed action at different distances from the monkey? There are neurons, as neuron 1, that encode action more strongly in the extrapersonal space, and others that encode action more strongly or selectively when they are performed within the peripersonal space of the monkey. With peripersonal space, I basically mean the space in which the monkey can act, can move, and can interact potentially with the target. These neurons change radically their firing rate in both sense, depending on their initial characterization, if the barrier is interposed between the experimenter's interaction with the target and the animal itself. Meaning that also mirror neurons encode not just pace, but a pragmatic representation of the action, as if, keeping with the initial interpretation provided for canonical neurons, the representation of the observed action of others has a different meaning if I can or cannot potentially interact with the agent. So, summarizing the findings that I presented so far, there is no categorical distinction between the so-called canonical and mirror neurons, so basically between neural representation of object and action within the parieto-premotor system. Premotor neuron responding to observed action can also respond to object and to a variety of other visual stimuli, and even to nothing if the task contingencies suggest that there could be an action in a given phase of the task. We have published this data in 2014. So there is an extreme flexibility in the functioning of the system, a flexibility that is basically matching the flexibility of our motor system in planning and changing our motor repertoire. Both object and action visual responses are mostly constrained to the peripersonal space, as we have been able to demonstrate with the different barrier tests that we performed both on object-related and action-related responses. And mixed representation may share functional pragmatic relevance wherever they appear within this larger network of areas. One interesting thing of the monkey neurophysiology is that you can actually inject neural tracer in specific brain region to characterize them in terms of connectivity. And we did so in the ventral premotor cortex, finding the surprising result that action observation response may occur independently of direct projection from STS. If you study mirror neurons in the inferior parietal lobule and you inject tracer, you don't find projection from the STS. Where you did find, actually, we did find in projection from the STS is after injections in the anterior intraparietal area IP, where we found a variety of visual responses to manual action. And this leads us to suggest that AIP could actually act as a hub for distributing information, for routing visual information into the network. And we also did the same test in the RF6, providing a sort of binding evidence that all these nodes of the network are actually interlinked one with the other. Exactly in area 6, there were findings in 2010 that enriched considerably our view, our idea of this system. So from the Yoshida and Isoda lab, they demonstrated the existence of neurons that were already characterized in the seminal paper on mirror neurons, but basically neglected as mirror-like, so mirror but not too much, that were basically tuned and selective for the observation of others' action. So there are neurons within the motor system in motor areas, we show them in F6, in F5, and even in AIP, that are selectively encoding others' action. I had another piece of evidence. From bird studies, we know that there are neurons in a region of the bird's brain that is homolog to the basal ganglia of humans that have a very precise audiovocal mirroring. So this triggers us, no time for this, this triggers us to look at the neurophysiological properties of the putamen nucleus that is connected actually with most of the nodes of this network. You will see more data in the poster session of tomorrow, poster 32, where we present extensive evidence that we are just preparing for publication, in which we could show that there are indeed neurons responding not only to action execution, but also to action observation in the putamen, and most interestingly, that the visual information itself is neither sufficient nor necessary, while the possibility to interact with the object seems to be, because if we do the barrier test in the putamen nucleus, it happens exactly what it happens in the cortical motor system. And if we create an observation condition in the dark in which the monkey basically knows that there is an action performed by the experimenter but cannot see anything, the neuron still responds. So the visual information is not necessary, but it must be pragmatic. The monkey must know and must be aware of the fact that it could interact with the target and with the experimenter. Another recent evidence is that part of these so-called mirror neurons in the cortex could be probably interneuron, inhibitor interneuron, which is something very far from the concept of neurons leading and driving our motor execution. And just to conclude, I would like to show you one of the video of the original studies carried out in the premotor cortex that I will show you 2 times, as I typically do with students in my lessons. So this is the video. Okay, stop it here because I'm scared by the detachment of the microphone. So here is some picture from a paper published by the Roger Lemon group where they provided evidence of exactly this type of neurons, so suppressed mirror neuron. That could be a mechanism for preventing the unwanted motor output during action observation. I'm pretty sure that most of you have appreciated what was happening while Giuseppe Di Pellegrino was grasping the object, covering it with the cap, showing that there is a selectivity for the end-object interaction in which the monkey looked at this phenomenon, but try to look at it in this way. So I'm pretty sure that you have noted that based on the suppression of the discharge, you could predict when the monkey is about to start its own action to grasp the target, something that you probably did not notice when focusing on the action of the experimenter, so on the other side of the video. But if you focus on the animal, and if you focus on the variety of neuronal properties that we could describe in recent years in this same region, it seems that the extended cortical grasping network is encoding others' action as well as physical object for the same purposes and with the same criterion. So to enable orchestrating the selection of one's own action. That could be a critical aspect for predicting other behavior and deciding when and how to respond. What we basically propose to be a sort of social affordance, not to talk about a different type of affordance, but to emphasize the fact that affordance are not provided just by the physical object as in the classical Gibbsonian theory, but they can be provided also by others' action, biological motion, that is basically the stimuli with which we have to deal with for the most time of our life, for our daily life, and even, according to Gibson, the most complex object of perception that animals have to deal with. So thank you very much for your attention. I hope I've been in time. I don't know if you want the microphone. Oh, perhaps, yeah. Can't do myself.
That was a great talk. Really enjoyed it. I wonder, and I'm sure you've thought about this, how much the modulation of the cells, particularly the pragmatic aspects, depend upon the training regimen, that the fact that you've trained the monkey to actually interact with these objects. And
considerably.
I have another question too, but okay. I'll let you answer. You answer that one first.
Okay. So considerably, in the paper we published in Current Biology in 2014, we basically show evidence of inaction neurons. So neurons that discharge during action observation, but that also discharge in a sort of agent-specific manner when just the experimenter refrained from acting because there was a cue that the monkey was overtrained to interpret as predictive of no action. So in the dynamic, the profile, we had neurons firing earlier and later with respect to the no-go signal. They maintain the same timing, the same profile of discharge during execution of the action and during inaction. And another set of neurons did so during the motor task. So during a motor task, it is reasonable. You have neurons that can contribute, maybe they are inhibitory interneuron, they can contribute to suppress the output when the instruction is not to go. But there are a set of neurons that do the same during action observation. So it means that the same plasticity mechanism that works for enabling us to act or refrain from acting in the motor task are working also during the observation. So these are even not visual response because there is no vision. This is an internally generated response that may depend on projection from the prefrontal cortex, so a sort of top-down mechanism that recruit motor plans when complex and learned contextual information are there.
Yeah, so my other question, it's kind of related to the barrier thing, but perhaps requires more decision-making on the part of the cognitive decision-making on the part of the monkey, where if you have objects that are not graspable by virtue of their size or the fact they're affixed to something, do you find that the system deals with graspable objects differently than objects that are clearly outside that domain?
Okay, the general response is that I don't know because we did not directly test this. What we did is to test, as I've shown you in Area AP recently, object presented on a screen. And this is radically different with respect to presented object physically and within the space of the monkey. Even the same object presented far from the monkey, which is head restrained, you should not forget that. What we are doing in the last 5 years is to focus on neurotological approach to study this monkey freely moving for exactly these two reasons. First one, most of what we see with trained monkey in this system is basically somehow, and we cannot say how much, now we can, a reflection of the training. Yeah. Or of learned plasticity mechanisms that somehow shape the way in which the neurons fire. But of course it was unavoidable. This was the only one way we had to do this kind of research. Now we are trying to shift to more ecological approach to see and compare how much the properties actually change because of this. Right. And the other point is that presenting real object that can be grasped, that the monkey can interact with, is radically different than presenting them on screen. So I don't know with more complex or non-graspable object, but this would suggest that the capacity to pragmatically interact with them is a crucial factor for at least activating neurons in the motor system. I don't know in visual areas.
Thanks very much.
Other questions?
Yes.
Thank you. Beautiful talk. I have a question regarding the point you made at the very beginning of your talk. That
regarding, sorry?
The very beginning of your talk, when you made the point that it's not enough to look at individual neurons, we need to understand the network. Yep. And I think you made a good point why that makes a lot of sense. So I was curious, do you think this is the network as it is, or how wide do you think we have to think this network? Are there additional areas?
Well, of course, the ideal thing would be to study the whole brain, as one could basically do with fMRI, for example. But the problem is that, as you know, the temporal and spatial resolution are radically different. So what we are trying to do, there are two opposite tendencies in the neurophysiological literature. One is to use, for example, neuropixel, highly dense probes that enable you to sample a huge number of neurons and to have a very specific description of a very small part of the brain. So in the extremely small. The other is to use, which is what we are preferring right now, matrix of probes to cover several millimeters of cortical convexity that, as in the approach used by Scherberger and many others, can allow you to sample activity simultaneously from different brain regions. And in this way, you actually have a more wider picture that can make sense of phenomena that if you look at the single area, for example, neurons that completely change its tuning from object representation to action execution. It has no sense, it is not canonical at all, and it makes it difficult to understand what's going on. But if you look at another area that is picking up this information and keeping in the circuit the stability of object presentation, then everything makes sense and can also account for how the brain is using these mixed representation and population code to gain stability and providing a more robust encoding strategy, more robust to perturbation and more robust also to lesion.
As a brief follow-up, out of curiosity, are you considering or are you planning to also pick up areas from the more pragmatic ventral stream, or?
What do you mean, for example?
Well, for example, areas from the so-called third lateral pathway, for example.
It's technically challenging. I've just read a paper yesterday that was suggesting me to do that, but I should calm down my enthusiasm because from a technical point of view, it's very tricky. And there are a few labs that do that, and so we should first learn how.
Thank you.
Thank you for your talk. So I had a question going back to the inactivation studies that you referenced in passing, and my memory was that those early mus studies by 𝗟𝗂𝗰 and 𝗟𝘂𝗰, that they also observed that the animals exhibited peripersonal neglect contralateral to the side of the inactivation.
Or extinction.
Or extinction, which is not misaligned with your argument. And I was curious if, well, first of all, what your thoughts are with respect to that. Is it truly an attentional deficit? And second, if you were to put a transparent barrier in front of the animal under conditions of inactivation of these regions, does the neglect actually recover? I mean, is the neglect a result of the possibility of there being an action, and when you take that away and you just let it be a truly perceptual response, does the animal no longer exhibit the seeming attention deficit?
I think that these kind of deficits mostly depend on the motor side. So you see a neglect, but not necessarily because it is a perceptual neglect. So far, there is very few evidence of the possibility, for example, of creating a neglect, which was in the 𝗟𝗂𝗰 study, not in the 𝗟𝘂𝗰 one, from parietal lesion, which was what is what we would expect if the system really mimic the organization of the human brain. There is, of course, no lateralization, so you can produce neglect from or an extinction for the contralateral space, regardless of which hemisphere you interfere with. But the point is that it's much more stronger and much more evident, rather than in Fugazi studies, in the earlier studies where they make a lesion in the ventral premotor cortex, and particularly in the arcuate sulcus, which is the region, the only one region of the premotor cortex, except F4, that has a corticospinal output. So the region which is more close to the output in this sector is the one that in the monkey produce more clearly this kind of deficit, which does not probably depend on the way in which the visual information is transferred to this region. So it's not something that can disappear from a perceptual point of view, because it is something that really depends on the capability of the animal with this region intact, but not with this region lesioned, to transform the position and the presence of the object into a motor plan directed to it. So here we are after the perceptual side of the story. We are in the motor domain. And this is why I would not expect a big change, unless the fact that if you interpose the barrier, maybe the object can be seen, but you have a problem from a behavioral point of view, how to demonstrate that the monkey can do that or can perceive it. Maybe with a task specifically designed to signal that the monkey is perceiving the stimulus. I would expect in this case that the monkey is perceiving the stimulus, but is not capable or not willing to direct the action to it. I don't know if I responded. But it's an hypothesis, of course. I don't know. That's definitely honest.
I just wanted to follow up on that very last point you made about if you lesion the arcuate. I mean, the frontal eye field is buried in the middle of the arcuate. So are you really sure that that statement's true?
They were. I didn't read myself. They were. They claim to be.
Okay. The frontal eye field is a critical structure for visual attention. And, you know, sort of it's at this nexus between vision and motor.
Studies, they did both. So if we believe, as I believe, that they actually did it, because it's from an anatomical point of view, when you open and you see the arcuate sulcus making a lesion anterior or posterior to it, is pretty obvious. So I'm inclined to think that actually there is this dissociation, because they could observe it also after the lesion of the frontal eye field.
Hi, thanks for your talk. I have a question about, I was thinking about the barrier and what the barrier means to the monkey, which, right, as you pointed out, is an action they can't do. And so what I'm wondering about is an action they don't want to do. So if you had a value associated with different objects, namely if you had, through conditioning, taught that some objects were highly aversive, maybe they would get shocked if they touched the object, or it's a food they don't want, or something like that. And so now they're seeing an action on an object that they could act on but don't want to act on. Would you get a similar kind of response, do you think, and at what points in the network?
So if we separate the two things and we focus on action observation, there has been a study by 𝗟𝘂𝗰 and co-workers that associate a specifically different value to different objects, and they show that actually this is sufficient to change radically, even from 0 to all, the response of apparently mirror neurons of the premotor cortex. So I would be inclined to respond absolutely yes to this claim. Of course, this is, has to do with action-related responses. Very likely this could happen also if you do the same with object-related neurons, because we have been able to see there is no such a huge separation between the two domains.
Thank you.
So I'll get to ask a question while you guys in the back think of questions. Can you hear me? Yeah. Thank you for a brilliant talk. I'm always fascinated by talks about the brain, because I don't understand much of what is said. But there was one thing you said that I found really striking, and that is that a neuron responds to an incredibly complex cognitive process. What is the theoretical claim you're making? Are you saying that a single neuron is capturing the kinds of claims you're making about intention and so on?
No.
Then what is the claim you're making?
So it depends on the point of view. What we are dealing here, I don't think even that you can claim that a single area or a single voxel that differently activate in an fMRI experiment is capturing or dealing or causally doing something cognitive, let's say, or even perceptual. It plays a role. There is centrally a neural machinery in there that is playing a role, that is doing something that enables the subject to express the cognitive or the perceptual process. I'm pretty convinced that when we reach a high level of description of this phenomenon, we are not dealing with single cell, with single voxel, or with single areas. We are dealing with the emerging properties of a circuit that implement a function and in which each single node plays a role in building up this kind of function that we can test from a behavioral point of view, of which we can find evidence, and we can try to correlate the individual neurons' properties to this phenomenon. Ideally, being capable of manipulating the single neuron activity to see what kind of perturbation we can exert on the properties. But first of all, we need a definition of what we are studying, which has been one of the main problems of this literature. When dealing with high-order cognitive properties, especially in animal models, you need to have very accurate operational definition, because otherwise there is a confusion on what you are studying. And the plethora of terminology emerge dealing with potentially the same process that is then difficult to understand who did what in this domain.
So is there some way then to partition the theoretical claims you want to make about what an action is or what the intentions and so on?
I never talk about intention.
Sorry?
I did in the past, but I didn't talk about intention here today.
Well, implicitly in terms of not intending, if you put a barrier, then the question is why is the monkey not reaching? Okay, so the intention to act only manifests itself when in fact there is no barrier.
Okay.
So the question then is, can you, I know it's an unfair question, so is there an attempt to come up with some kind of explicit set of claims about how we can partition whatever theoretical machinery you want to postulate so that we can map it onto the brain? When you say this neuron is this, I don't know what it means. What do you mean this neuron responds to this? It responds to an incredibly complex set of things that you point out. So it's part of some complex network. But can we envision how we can assemble a whole bunch of neurons to come up with an explanation of the things you want to explain? Or is that something that we should give up on at this point in time?
I think so. There are elegant studies. There is a recent one that comes to my mind, is one on which I based my last 𝗟𝘂𝗰 consolidator proposal that has been funded, and we are working on this, that show that, for example, if you focus, and this is why I mentioned, of course I didn't have enough time to develop the argument, if you focus on neurons that have exquisitely other related properties, they are in a motor area. They encode other related stuff. They do not respond while the monkey moves. So why they are there? And they are intermingled with neurons that have nothing to do with other. They do not discharge at all during other movement, but they discharge when the animal moves itself. It has been done in mice with calcium imaging, recording from two simultaneously interacting animals from their two brains. And it has been shown, for example, that the coupling between self-type neurons in the brains of one animal and other-type neurons in the brain of the other animal is the one that maximizes the coherence, and the coherence is predictive of the animal behavior. As if there is a machinery that is not magic, but that takes as an input the observation of the other's behavior to translate this behavior in neural terms in the planned and finally executed motor outcome of the receiver brain. So this is something that can enable us to link the single cell description of a complex behavioral phenomena in mice. In this case, we are trying to do that in monkey, and to derive an explanation of the behavior that can also be causally investigated with DREAD, with different genetic technologies, and so to understand how things happen linking the network to the single cell mechanism. But the point is that you need definition, as I said before, and you need to focus on a specific aspect to study.
No other question, I'll ask my last question. So I apologize at the beginning of my ignorance about brains, but there was this sort of idea almost that when we look at the brain, what we find is that there are these basic small units, and that we are smart enough to show how the combination of those units makes possible whatever idea we have in mind to express. So in the area of visual perception, we go from low-level features to richer and richer and richer features. Now, you've taken us well beyond that. You've taken us in the world of action, and it involves all kinds of aspects, not just the visual shape of things, not only the kinds of motor acts you can perform, but also whether you want to act or not, how you can act. So now the notion of a neuron, in this case, in this world, is complicated because we don't have any longer, or what seemed to be the simple idea of units that all have the same properties, and that we deal with various kinds of hierarchies. The hierarchies here are incredibly complex, and as you point out, they don't reflect the firing to a particular object only, but the context of the object.
Sure.
How it connects to other things. We are far from having a theory then of how the brain performs any of these things.
We have a lot to do.
So when we label this unit X, okay, what we're saying is that it is part of some mysterious thing at this point that makes it possible for us to act on the world. It seems to be more of a mystery than an explanation at this point.
I'm absolutely agree. I mean, there is a lot of work to do, but I really think that trying to tackle the problem from different point of view, from different perspective, from different level, is the only one way we can deal with such a complex machinery, because we cannot deny that these levels are there, that the brain is not made of individual circuit, but they can be decomposed. An individual neuron was, and emerging properties take place from all the levels of description of the system. So the challenge is to link these levels, not to discard or to focus on a single one, in my view. And of course, from a technical point of view, it is difficult because it would imply to have at the same time the possibility to tackle the problem from all the different levels and see how they interact, which is what we are trying to do since several years, also from a technical point of view, but I agree, it's tremendously challenging.
Well, I want to thank the speaker for this brilliant talk and for, I think, doing a wonderful thing for us, those of us who are cognitive scientists who are trying to look at cognitive neuroscience to help us to realize that we're in the same boat. That is, until we have a clear functional theory in terms of what we are trying to explain, and that requires a cognitive science perspective, I think that the neuroscience will just be floundering. We need to combine those two areas, and I think this talk beautifully illustrates that. Thank you very much. And now,